Ever wonder how your cells know what to do? It all comes down to a simple flow of information that controls everything inside. Let's dive in. Central dogma is the process by which genetic information flows in living cells from DNA to mRNA and then it translates into a protein. The conversion of DNA to mRNA is called transcription and the conversion of mRNA to a protein is known as translation. When we look at the structure of a chromosome, it's essentially the result of highly organized DNA packaging. DNA itself is the basic structural molecule which consists of two purine bases A and G and two pyrimidine bases T and C. They form complementary base pairs, 2 hydrogen bonds between A and T and 3 between C and G. The first process is transcription. Transcription is how a cell copies DNA into RNA. RNA carries the instructions to make proteins. The enzyme RNA polymerase reads the DNA and binds to a promoter, getting ready to start transcription. It unwinds the DNA strand. One of the unwinded strand is known as the template, non-coding strand, and the other is the coding strand. The RNA polymerase moves along the template strand, reading it from its 3' end to 5' end, synthesizing a new strand in the 5' to 3' direction. Once the messenger RNA has been synthesized, RNA polymerase releases both the template strand and mRNA. It then detaches from the DNA strand, allowing it to retain its helical structure. The DNA now turns back to its original form, ending the process of transcription. The mRNA is formed in the cytoplasm in prokaryotes, while in eukaryotes, it forms in the nucleus, and after further processing, enters the cytoplasm. So, the next part, translation, can take place in the ribosome. In eukaryotes, ribosomes have 40S and 60S subunits, while in prokaryotes, they are smaller with 30S and 50S subunits. The small ribosomal subunit binds to the mRNA to initiate translation. Translation begins as a tRNA molecule binds the start codon on the ribosome. A second tRNA matches the next codon, and their amino acids join by a peptide bond. The mRNA then moves forward so that another tRNA molecule is attached. Only 2 tRNA molecules can bind at a time, so when the first tRNA leaves, the other enters. At the end of the mRNA is the stop codon that is responsible for stopping the translation process. Once the translation has been stopped, a newly synthesized amino acid chain is released, which undergoes further modifications. It undergoes various post-translational modifications, such as phosphorylation, in which a phosphate group is added to the OH-containing residues. Glycosylation, in which carbohydrates are added for proper structural and functional working. Ubiquitination, in which a small protein is covalently attached to target proteins. Acetylation introduces an acetyl group to lysine residues for regulation and expression. Methylation involves adding a methyl group to a molecule, which acts as an essential molecular switch. Lipidation, in which a lipid group covalently attaches to another molecule, increasing its hydrophobicity to anchor the cell membranes. Proteolysis involves the hydrolysis of peptide bonds to break down proteins to serve as a permanent molecular switch. This journey ends with the amino acid chain now being modified and folded into a protein, ready to perform its function. The central dogma of molecular biology has been a hot topic for scientists all over the world, and new discoveries are being made in this domain with each passing day.